Hi, and welcome back to our continuing short video series on an introduction to phylogenetics for primarily life sciences students taking an introduction to bioinformatics. So let's talk trees, in particular, phylogenetic trees. What is phylogenetics? More broadly, it's basically written here. It's the study of evolutionary relationships. And we can use lots of different types of data, genetic data, quantitative traits, all sorts of characters, and morphological data matrices. So it's not restricted just to molecular data, but the subfield of molecular phylogenetics is that which is really focused on molecular data. And by molecular data, we mean DNA and protein sequence data. And of course, most of that is going to be DNA sequence data, or translated to proteins, but experimentally DNA sequence data. The result of doing phylogenetics is generally a tree, along with some other information, but information to describe the tree. The tree is the central piece, and this is a branching diagram, and the idea of the tree is that it shows the evolutionary relationships that we infer from the data, and those evolutionary relationships in the form of that tree is referred to as a phylogeny. Why do this? Well, one key principle is that trees themselves, over and above multiple sequence alignment and some of the sequence analysis approaches that we've already studied, phylogenetic trees, that structure that results, is really empowering. It helps us think more clearly about the relationships between species or between sequences when we're doing trees of gene families and so on. Secondly, we have to keep in mind that when we work with phylogenetic analysis, when we work with sequence data, most of the time we only have sequence data of living species or relatively recently extinct species, and we infer aspects of ancestry. So this is different from, say, working with morphological characteristics from fossils, because not all organisms leave fossils, but of course that allows characterization of extinct organisms. In the case of DNA sequence, we're mostly dealing with existing organisms, but to the benefit of that analysis, all organisms have DNA, so we can generate trees that link together organisms that are very distantly related. What do these trees look like? Well, there are lots of different ways of displaying them. On this slide, there are just two shown, but they share some common characteristics. Let me just read here from the left, or start from the left. One characteristic is the internal node. So it's shown here and it's shown here. This is a point when one lineage in the tree branches to form typically two branches, and it's that branch point, that node. And that generally represents some ancestor, where it's the ancestor of the later ones going towards the leaves of the tree. The external nodes, that is the leaves of the tree, generally represent the existing species, the known species, the living species. So they represent generally our data when we're dealing with molecular phylogenetics. So these are external nodes. They're also referred to very often as leaves or the twigs, but leaves typically of the tree. And there are also, you may notice, there are different links of these branches. And so sometimes this might be just for convenience to separate the nodes and the leaves. But in other cases, they will represent evolutionary distance as measured in some way, or as estimated in some way, different types of measures of evolutionary distance. So by that, this distance here is larger than this distance here. So that means more evolution has happened from the common ancestor of A, B, C, and D, and E, which is here, towards the A-B split, then happened towards the split of E from C and D. So the branch links represent that. So these are two different representations. There are many more types of representations, but there's another distinction that's important, and that's a distinction between rooted trees and unrooted trees. So an unrooted tree, like the two shown over here, we can't tell the exact order, the order of evolutionary events in this kind of tree. We can't tell whether, for example, the common ancestor of all three of these, A, B, and C, arose, say, here and diverged towards B first and then towards A and C, common ancestor, or whether it arose, say, with the common ancestor of C diverging from the common ancestor of A and B first. We can't tell. And so that question of what is the most recent common ancestor of the data that's represented on the tree is not strictly answerable in the case of an unrooted tree because we don't know. But when the tree is rooted, as it is here, so we see this branch here, but that root branch may not always be represented, but we see the root represented here. The nodes, each of the nodes represents the most recent common ancestor of the branches that follow it in the model. And in a rooted tree, as in an unrooted tree, the links of the branches may be meaningful. Sometimes, not always, sometimes we can see an unrooted tree and it looks suspiciously like a rooted tree, and we might have to look at it a few times or look at the caption in order to tell whether the tree is actually rooted or not. So here are a few more terms. One is a bifurcating tree. So most trees, rooted trees, will be drawn as bifurcating trees where two descendants arise from every interior node. So each common ancestor splits into two descendants, or each internal branch splits into two progeny branches. A dendrogram is a general term for all of these types of diagrams. So a phylogenetic tree is a type of dendrogram, but not all dendrograms are phylogenetic trees. So sometimes we'll see dendrograms drawn just in the course of exploratory data analysis, hierarchical clustering, and so on, that have nothing to do with phylogenetics, but the tree is a very useful structure for organizing the data in data analysis. Those are also dendrograms, of course, but they're not phylogenetic trees. When we start to work with phylogenetic trees, there are many different types, and some of the different names represent this. I only have two on this slide, but they represent different meanings for the branch links. So in the case of a phylogram, the branch links represent the amount of character change. In the case of a chronogram, the branch links represent some aspect of evolutionary time. So there are these different types of trees. And there are other ones as well, but those are just sort of starting terms that might be useful. Here's an example of a phylogenetic tree that is unrooted, but the branch links are meaningful. So here we can't really tell in this case, where the most recent common ancestor was in all of this. But we do see these branches. So these are often drawn in this kind of fan-like form where they're just trying to separate the leaves so that the individual, so that they're spread out well enough, although you can see that they're not quite spread out well enough here. The same tree can be redrawn as a phylogram. So in this case, the branches are drawn as a phylogram, the branch links are drawn as a phylogram, but this is actually the same tree here. So it's the same tree construction method, it's just drawn differently. And notice, in this case, it's not bifurcating. We see these three branches going off of here, but we can't really tell where the most recent common ancestor is, even if it is along one of these. We can take the same data, the same sequence alignment, and draw a rooted tree using one of the rooting methods. In this case, it's rooted using something called a molecular clock. And you might notice immediately that this tree is bifurcating. Every branch goes to two branches. You might also notice in this tree that all of the leaves are the same distance from the root, which is characteristic of a tree generated using a molecular clock. And you might also notice that the root itself is not actually drawn. So this tree, if you were to look at it, you might not tell, you might not immediately see that it is a rooted tree because there's no root line coming out here from the left. Nonetheless, this is a rooted tree. So how do we actually do it? How do we go about generating a phylogenetic tree? Well, the first step is deciding what sequences to include, because of course that's essentially where the tree comes from. We then align those sequences and generate a multiple sequence alignment, and we might need to do some editing and trimming of gaps and so on after that. Then we use one of a number of tree estimation methods that we'll go into later. And then if we want to, we root the tree and identify a root that we can use to draw a rooted tree, and then we draw it, and we draw aspects of that tree that allow us to interpret it, allow us to interpret its stability, and so on. Let's go through those steps a little bit. So the first part, how do we decide what sequences to include? One way to think about this is that it is the same as choosing the tips, the leaves of the tree, because each of the sequences that we include eventually becomes a leaf in the tree. So if we think of it that way, it might help us choose the sequences that we want to include depending on our task. This can be very time-consuming because we might need to do a lot of work to decide whether to include one sequence or exclude and so on. How do we decide? Well, we have to think about the problem. We're trying to address some problem, trying to understand the answer to some question, and the answer to what sequences to include is really just related to the question we're trying to answer, nothing more. How do we do it? We generally do searches. We take a sequence that we know we want to include and we search to find related sequences according to the criteria that makes sense. So a common way to approach this is to simply use one form of BLAST algorithm or another to search for sequences that are related to the sequences that we know that we want to include. Okay, that's step one. The second step is aligning the sequences. So any of the standard MSA programs, Clustal, Muscle, T-Coffee, ProbCons, so on, any of them can be used depending on what you're trying to get at. You may need to edit the alignments. So typically what happens is you'll want to trim off the ends if there are variable length ends of the alignment, so trim off the ends that are variable. You might need to trim even internal sequences that are gapped. There are reasons for doing that because of the way the tree inference algorithms work, where, say, long ends that are only on some of the sequences can bias the results. They can sort of pull the results towards certain sequences. In particular, when you're choosing an MSA program and you're deciding how to run it, you have to take into account all of the caveats of MSA. So if you're using Clustal Omega, then you have to recognize that it's a progressive algorithm, and so any of the mistakes that get introduced early in the algorithm will be retained and persist through the entire algorithm. If you're using Muscle, then there's some iteration that allows at least exploration and correction of some mistakes. If you're using a consistency-based method, there'll be those considerations applied and so on. So you have to just keep in mind all the caveats of MSA and use those wisely. So here's an example of a multiple sequence alignment. In this case, it's a protein alignment, as you can see. And a key thing to remember in this is that the purpose here, when I say here, I mean for drawing phylogenetic trees or deriving phylogenetic trees, the purpose of multiple sequence alignment is to align characters, that is, the letters, in the same column if they are conserved, homologous, and subject to selection. So in this case, we can see all these cysteines, this phenylalanine with a serine substitution here, this methionine with a glutamate substitution here, and so on. So we can see some totally conserved amino acids. Some amino acids, say these are mostly basic amino acids, lysines and arginine, but they're subject to selection, and the substitutions are arranged on those homologous positions subject to selection in a single column. This is really the only purpose of multiple sequence alignments. It's to put those homologous positions of homologous sequences in the same column so that the columns are a representative of the forces of selection. Now, in terms of estimation methods, in this video, we won't go into detail on estimation methods, but there are two broad categories to estimate trees. The first is distance-based methods. So what we do there is after the multiple sequence alignment, we take those aligned sequences and we convert those sequences into a distance matrix, into distances, and those represent some form of pairwise differences between sequences. And this will depend on, say, the substitution model that we use and so on, how those distances work. And then we use just the distance matrix and infer a tree from that using one of two methods, either neighbor joining or UPGMA. A distance matrix method, so distance-based methods, yield a single tree. They can yield more trees in Bootstrap, but yields a single tree from the data itself. Character-based methods are another class of methods, and in those cases, after the alignment itself is used instead of a distance matrix. And it's used by taking each position, treating it as a character, and comparing those characters. Of course, this means that what's on the previous slide must be true. The homologous position must be aligned. So in character-based methods, most of the time, many trees are generated and they're compared to each other against some score. And because of that, they can be slower. There are also far too many trees to enumerate all of them. So this is an example, 10 to the 195th trees for 100 species. So that's just way too many. And so in order to solve the problem using character-based methods, various forms of search, hill climbing, and so on are done. And we'll get into those in a later video. And finally, rooting a tree. How do we root a tree? Well, there are a bunch of methods. These are the most common. The most common is what's called the outgroup method. So there, if we know that if we're looking at, say, evolution within a particular set of species, then we can take a sequence that's outside of that. So we know that that sequence diverged from all the sequences that we are interested in before those sequences diverged from a common ancestor. And then we just we infer a tree, an unrooted tree, however we like, but we know that the root needs to be between that outgroup and the rest of the sequences. And so we just remove the outgroup sequence and replace it with the root. Another method is a midpoint method. So in the midpoint method, we draw a tree and we decide that the most evolutionary change, so the two leaves that are furthest apart from each other, should represent the most divergence, and we put the root in the middle, halfway between those two. Another method that would be useful if we're looking at ancient gene duplications is we just place the root between paralogs of gene copies. And then finally, this method, UPGMA, is a distance-based method that assumes a molecular clock and yields a root by virtue of that assumption. So I hope this is useful in introducing some of the ideas of phylogenetic trees, and we'll continue this with discussions of some of the specific tree inference methods. See you in the next video.